Imagine a future where failing kidneys could literally repair themselves, potentially ending the need for dialysis altogether. Welcome to This Explainer. Today, we're putting together a comprehensive video summary of the renal MSC blueprint, diving deep into the clinical realities, the mind-bending biological mysteries, and the massive future paradigm shifts of mesenchymal stem cell therapies for chronic kidney disease. A total revolution in cellular healing is happening right now, and the data is pointing us in a direction almost no one saw coming. So let's jump right in. We're going to visually anchor our discussion today by meticulously walking through this blueprint. We'll map out exactly how these treatments are evolving from our current, somewhat brute force status quo, all the way to incredibly sophisticated next-generation strategies. Our roadmap today covers 4 main stops: clinical realities, the delivery paradox, paracrine mechanisms, and finally, the future of acellular therapy. Let's get to it. This executive synthesis perfectly teases our entire narrative arc. We're going to move from validating early clinical safety right through a massive physical bottleneck known as the pulmonary trap, and finally arrive at the secretome paradigm. It's a fascinating journey, really. We're shifting from assuming these cells act like physical building blocks to realizing they actually operate a lot more like remote pharmacies. Section 1, Clinical Realities and Efficacy. When you look at the clinical parameters from 18 completed phase 1-2 trials, one thing becomes crystal clear. Therapeutic efficacy in chronic kidney disease isn't just some simple on and off switch. It's highly variable. Efficacy depends intrinsically on the specific cell source, like whether it comes from the umbilical cord or bone marrow. It also relies heavily on strict GMP imperatives. That means rigorous manufacturing standards are absolutely crucial to verify the identity, potency, and sterility of the cells before they ever even reach a patient. In the current clinical landscape for primary indications like lupus nephritis, the standard systemic intravenous dosages are frankly massive. We're talking hundreds of millions of cells per kilogram of body weight. But the truly remarkable part here is the timeline. Clinical improvements don't just happen overnight. They track consistently across follow-up windows of 3, 6, 12, and even up to 18 months post-infusion. Now, the crucial point here is the contrast between safety and efficacy. Rigorous tracking shows literally zero statistical correlation between common adverse events and the MSC products themselves. They are fundamentally safe. At the same time, the efficacy signals are incredibly strong. We're observing up to a 60% improvement in primary renal function markers. For instance, glomerular filtration rate, which is essentially the kidney's filtering power, goes up, while toxic byproducts like blood urea nitrogen are significantly reduced. Section 2, the delivery paradox and the lung trap. This brings us to a highly pointed, almost uncomfortable question. If the intravenously injected cells never actually reach the target kidney tissue in meaningful numbers, how on earth is the patient healing? Let's unpack this core delivery paradox. The original hypothesis assumed you inject MSCs into a vein, they travel systemically, find the damaged kidney, and graft into the tissue and physically rebuild it. Makes sense, right? But the clinical reality tells a completely different story. Because MSCs have high adhesion molecules and a relatively large physical size, they cause massive systemic bottlenecks very early on in circulation. Welcome to the pulmonary first-pass effect, often called the lung trap. When you inject millions of MSCs, about 99% of them get physically trapped in the 8 billion pulmonary capillaries of the lungs. The lungs effectively act like a giant physical net. With only a microscopic fraction of cells ever reaching the actual renal target, direct tissue engraftment isn't just unlikely, it's mathematically impossible as the primary mechanism of action. Section 3, Paracrine Mechanisms and EV Delivery Systems. So here is the huge breakthrough realization. MSCs are not physical bricks meant to rebuild tissue. They're actually remote factories. Once they get trapped in the lungs, they start secreting roughly 1,500 distinct paracrine components right into the bloodstream. They fundamentally alter the systemic microenvironment by upregulating healing cells like Tregs and M2 macrophages, while simultaneously suppressing the pro-inflammatory cells. They are literally healing the kidney remotely through immunomodulation. But there's a catch with this remote delivery. If the trapped cells simply release soluble proteins, those proteins would face massive, rapid dilution in your 5 liters of blood plasma. The concentration would drop to almost nothing. Instead, the secretome relies on targeted extracellular vesicles, or EVs. Think of these as protective lipid envelopes that pack crucial healing instructions inside, completely bypassing the lung trap and the dilution problem to deliver stable, mechanical healing directly to the kidney. The temporal reality of this whole process is fascinating, mainly because it's not instantaneous. For the first 24 hours after injection, the cells go through a massive stress phase. They are completely dormant, basically just trying to survive being trapped in the lungs. It isn't until days 1 through 3 that this biological engine actually turns on and begins the active secretion of those EVs. And this unlocks a massive clinical insight. Because tissue repair requires sustained signaling, single injections just aren't enough. We absolutely need multi-dose intervals to keep this remote healing engine running. Section 4. The future of acellular therapy. All of this raises a massive strategic question. If the secretome is the actual cure, do we even need the cell at all? Our current reality relies on live cellular engines, with variable survival rates that just get constrained by the lung trap. But the future points directly toward acellular conditioned mediums. By synthesizing the plasma proteins and EVs directly in a lab, we can create a highly scalable therapy that totally bypasses the lung trap altogether. And that brings us to the strategic action plan for the future. First, we've got to refine the secretome by mapping the exact plasma proteins needed. Second, we must evolve our delivery mechanisms, perhaps using local renal arterial perfusion to entirely bypass that systemic lung trap. And third, we need to innovate our cell sources, utilizing iPSCs, which are essentially reprogrammed stem cells, to mass produce these targeted acellular therapeutics while keeping the patient's immune system perfectly safe. As we conclude our analysis today, the evidence presents a thrilling crossroads. We started by looking at live cells as the ultimate physical healers, only to discover their true power lies in the invisible, remote messages they send while physically trapped in the lungs. So, I leave you with this provocative thought to chew on. Are we on the verge of abandoning live stem cell injections altogether to finally unlock the pure, highly scalable potential of the secretome? Think about that, and I'll catch you in the next explainer.